Antoine Lavoisier's 1777 experiments on the respiration of animals and on combustion constitutes a pivotal contribution to chemistry and physiology. This work, meticulously documented and carefully reasoned, represents a crucial step in dismantling the phlogiston theory, a dominant paradigm of the time, and in establishing the foundations of modern chemical understanding. To fully appreciate its significance, we ought to trace its genesis, examine its core tenets, and assess its lasting impact. Prior to Lavoisier's investigations, the prevailing explanation for combustion and respiration rested upon the phlogiston theory, propounded primarily by Georg Ernst Stahl. This theory posited that all flammable substances contained phlogiston, a hypothetical substance released during burning. Combustion was conceived as a process of phlogiston escaping, leaving behind a dephlogisticated residue. Similarly, respiration was understood as a process involving the transfer of phlogiston between the animal and the air. This framework, while seemingly comprehensive, suffered from several critical limitations. It struggled to account for the observed increase in mass of many substances upon combustion, a phenomenon that directly contradicted the idea of phlogiston being a weightless or negatively weighted entity. Further, it failed to adequately explain the observed changes in air during both combustion and respiration. Lavoisier, a keen observer and skilled experimentalist, embarked upon a series of experiments to address these shortcomings. His approach was meticulously quantitative. Instead of relying on qualitative observations, he constructed precise apparatuses and methodically measured the changes in mass, volume, and composition of substances during combustion and respiration. These experiments, detailed in the 1777 work, represented a turning point in the history of science. Lavoisier introduced a level of rigor and precision that was unprecedented in the study of chemical processes. The theoretical crux of Lavoisier's work centered on the concept of oxygen, a substance he initially termed eminently respirable air. Contrary to the phlogiston theory, Lavoisier showed that combustion involved the combination of a substance with this component of air, which, he determined, was approximately 1/5 of the atmosphere. Specifically, he demonstrated that during combustion, a substance combines with oxygen, increasing in mass. This increase in mass, Lavoisier argued, was due to the addition of oxygen, not the loss of any hypothetical entity. Similarly, demonstrated that in animal respiration, oxygen was consumed and converted into thick air, which is now known as carbon dioxide. Lavoisier's experiments on respiration, in particular, were ingenious. He designed an apparatus that allowed him to measure the oxygen consumption and carbon dioxide production of animals. He enclosed animals in sealed chambers and meticulously analyzed the air before and after the creature's respiration. By analyzing the differences in air composition, he was able to establish a clear relationship between respiration and the consumption of oxygen. He found that respiration, like combustion, was a process of oxidation, the combination of a substance with oxygen. Moreover, he established that respiration, like combustion, produced heat. This observation linked respiration to the metabolic processes occurring within the body and established that the body utilized oxidation for generating energy. The impact of Lavoisier's work extended far beyond the immediate refutation of the phlogiston theory. It laid the groundwork for the modern understanding of chemical reactions. He introduced the principle of mass conservation, a fundamental concept in chemistry. He demonstrated that in a closed system, the total mass of reactants and products remains constant, a principle that continues to underpin all chemical calculations. His work also provided a more accurate understanding of the chemical composition of air. He meticulously documented the proportions of oxygen, nitrogen, and other gases, paving the path for the gas laws and the development of pneumatic chemistry. The immediate impact of Lavoisier's findings was a slow, but undeniable, shift in the scientific community's understanding of combustion and respiration. Though some chemists initially clung to the phlogiston theory, the mounting evidence generated by Lavoisier, and later his collaborators, proved increasingly difficult to ignore. The precision and rigor of his experimental methods, coupled with the clear and compelling explanations, gradually won over the initial skeptics. The Royal Academy of Sciences, initially hesitant, would eventually recognize the profound importance of Lavoisier's work. The principles of mass conservation and oxidation would transform chemistry by providing a framework based on measurable elements and quantifiable changes. The enduring legacy of Lavoisier's work is colossal. His contributions transformed chemistry from a qualitative art into a quantitative science. He established a rational system of nomenclature, providing a language that enabled precise communication and facilitated the rapid advancement of the field. His work on the conservation of mass became a foundational principle influencing all subsequent chemical investigations. The understanding of respiration and combustion he established remains at the core of our knowledge of biological processes. The concept of oxidation, a direct outcome of his investigations, is fundamental to understanding cellular respiration, energy production, a chemistry of materials. Lavoisier's work also influenced later developments in physiology. His demonstration that respiration is a form of oxidation provided an essential link between chemistry and biology. Subsequent researchers, inspired by Lavoisier's approach, began to explore the mechanisms of cellular respiration in more detail. This research led to a deeper understanding of metabolic pathways and the role of energy in biological systems. His focus on quantitative analysis influenced the evolution of experimental methodology in biological sciences. The contemporary reception of Lavoisier's work was mixed. Early in his career, he faced skepticism and even opposition from some established chemists who were invested in the phlogiston theory. Supporters of phlogiston were unwilling to abandon the prevailing scientific model. However, the clarity and rigor of Lavoisier's experimental results, along with the power of his theoretical framework, eventually won over many converts. His work attracted the attention of other prominent scientists, many of whom became his collaborators. His use of quantitative measurements and careful documentation set a new standard for scientific inquiry. Lavoisier's methods, though initially disputed, influenced chemistry. His approach to experimentation, his emphasis on precise measurement, and his use of quantitative reasoning became models for chemistry. The systematic nomenclature he developed, along with his meticulous record-keeping, facilitated the standardization of chemistry. All these developments were vital for the advancement of chemistry. The work influenced scientists around the world. The specific individuals whose work demonstrably influenced Lavoisier were primarily those involved in the burgeoning field of pneumatic chemistry, such as Joseph Priestley and Henry Cavendish. Priestley's discovery of oxygen, which he initially called dephlogisticated air, provided Lavoisier with a crucial piece of the puzzle. Cavendish's experiments on the composition of air also informed Lavoisier's work. Lavoisier built upon their observations, expanding them. He was not simply a passive recipient of information. He was a critical evaluator, refining the concepts and framing a new understanding. His own experimental work, however, transcended the prior work, providing the complete picture and resolving the prevailing scientific disputes. The influence of the earlier researchers, while undeniable, was integrated and transformed by Lavoisier's innovative framework and the application of his precise measurement method. Conversely, Lavoisier's work directly influenced a cohort of subsequent scientific figures, such as Joseph Louis Gay-Lussac, who built upon Lavoisier's work on gas analysis. Gay-Lussac contributed significantly to the understanding of gas laws and the chemical reactions. Another significant figure was Humphrey Davy, whose research on the nature of heat and electricity was directly influenced by Lavoisier's work on combustion and respiration. Davy provided greater insight into the nature of chemical bonds and the role of elements in chemical reactions. The work of these individuals, along with many others, demonstrates the far-reaching impact of Lavoisier's contributions and the foundational role he played in the development of modern chemistry. Consequently, Lavoisier's work became a cornerstone upon which subsequent discoveries were made. It has served as a touchstone for research for chemists and biologists alike. In closing, Lavoisier's experiments on the respiration of animals and on combustion represents more than a scientific paper. It represents a revolution in scientific thought. By introducing rigorous experimentation, precise measurement, and a fundamentally new theoretical framework, Lavoisier transformed the study of combustion and respiration. He not only dismantled the phlogiston theory but also established foundational principles that continue to shape our understanding of chemistry, biology, and the very processes that support life. The enduring legacy of Lavoisier's work is a testament to the power of observation, the importance of quantitative analysis, and the enduring quest for scientific truth.